Okay. So I think we can start. Good morning. Audio video is okay, right? Any lag? There's only online people today, right? Online people. Any audio video lag? You can clearly hear me and see me, right? How about the board? Writing on the board, stuff on the board, all clear, right? Others, any issues? What about the issues you are having? Right? Hope all of you are safe and your loved ones are safe, right? Your property, I mean, property comes last, your life, and then your closest people, then your valued properties. Those are the things you need to protect, right? um, the institute is actually open today, but I thought it will be a sort of a headache for you and your parents depending on where you are. I think around this Nugegoda and Colombo area the weather is fine, right? But I think the people who are affected could be still recovering from the effects, no? So I thought of not troubling with them with the pressure of coming physically to the class, as well as this is not a mandatory session. Anyone who's not able to join will be given the recording, right? So anyway, I was planning to discuss this paper as online, purely as an online, because I need uh, I mean, I have limited time in my theory class time slot. But anyway, because there's no other option than to do it on our usual slot because of this unexpected weather situation and everything, right? So that's why we have come to this arrangement of discussing this paper online today, right? So the plan is, uh, I was going to give you marks today itself and then name the people who got the top scores. That I will do, but not today, right? Because there's going to be a highest scorer in the physical class and a highest scorer in the online class, right? So I will do that next week. Today it's going to be just the discussion. So join with the discussion and we'll quickly finish it as soon as possible, right? Because this was just a session that was done for you to give an exposure before your first term test, right? Exposure to sort of actual A level exam level questions, that standard questions before you went to the exam. Every week we do past paper A level exam questions, right? Even though you are still the first batch, right? First years, I do A level exam questions with you, right? Categorize exam questions with you. So it's not an excuse that you are still the first years, because in another 3 months almost, you are going to be second years, because the 2028 people are going to be joining the journey, right? So anyway, let's start with the discussion. I'm going to go a bit fast over the straightforward MCQs, right? I'm going to go fast over the straightforward ones because there's no use in wasting time with just theoretical sections, right? Just the theory parts if they are question, right? So first MCQ, hope everyone is clear and you can all see everything, right? Online people, only online people, right? Any issues? No, right? Okay. So first question, correct statement regarding issues and statements pertaining to biology is, right? First one, life on Earth formed around 3.6 billion years ago. What do you think? Right? I need you to interact with me a little bit, right? So that you don't fall asleep, especially in this cold morning. Was it 3.6 billion years ago? No, right? It was not 3.6 billion years ago, right? I think the new resource says it was 3.5 billion. No? 3.5 billion years ago, life on Earth formed, right? It is assumed there are about 1 to 10 million species. That is, what do you think? Is that the case? No, right? There's about 10 to 100 million. No? So, statement number two is also wrong. Consumption of natural resources causes environmental problems. Now you have to be careful. This one is, I mean, it makes sense. It generally makes sense. Yes, consumption of natural resources, particularly if you think about fossil fuels, Emma, right? If you're burning fossil fuels, it can cause environmental pollution, right? But because we are going, I mean, because we are a resource book based paper style, right? According to your resource, it says, I think, overconsumption, no? Right? It says overconsumption. Online people, can you see this edge? Yeah, right? Overconsumption of natural resources, right? Because if you use them in a sustainable way, there won't be much problems. There's going to be a proper balance, particularly if you take the carbon cycle. If you emit the same amount of carbon, if you emit the same amount of CO2 that is absorbed and that is evolved, right? You learn about the carbon cycle, right? If the amount of carbon dioxide evolved is hold on, it's a bit laggy. The amount of carbon dioxide evolved is equal to the carbon dioxide absorbed, then there won't be any imbalance in the carbon cycle. So, overconsumption of natural resources cause environmental problems. Sustainable food production is not based on these are, I mean, very obvious answers. Anyone who's not even studied can get these sort of statements. I mean, figure out these sort of statements. Heart disease is a serious health problem in which causes are not fully understood. General knowledge, right? Anyone who's not doing bio, maths can right? Heart disease is a serious health problem. People who are apparently healthy without any genetic history, තාත්තටත් නැහැ, අම්මටත් නැහැ, සියයටත් නැහැ, අච්චිටත් නැහැ heart problems, but they have. So, I mean, if you take pathologically, we have understood most of the basis for heart disease, but according to your resource, it's a the causes are not fully understood, right? So anyway, answer number 5. Okay, answer number 5. Moving on. Following are the steps to be followed when a specimen is observed under the compound light microscope. The response with the correct sequence is right. Online people, is there a lot of noise? Is there a lot of background noise? Yes, I think no, right? My voice is clear, right? Okay. Right. Following are the steps to be followed when a specimen is observed under the compound light microscope, right? Place the slide on the stage of the microscope facing the cover slip on top. අන්තිමටම stage එකට mount කරන්නේ. So whatever A has to be sort of last. Uh, cover the specimen with the cover slip is sort of before that. No? අපි cover slip එකෙන් cover කරන්න before we mount it on the stage, no? So whatever the stage with the cover slip has to be before mounting. Place the thin epidermal peel on a water in a petri dish. Okay. So that is usually sort of the first one. No? අපි onion epidermis peel එකක් after we have using a razor blade, once we have taken piece of onion epidermis, we put it in water to prevent what? To prevent it from drying out. So probably C is number one. Then D is remaining. Transfer the specimen into the drop of water placed on the center of the glass slide. Right. So from the petri dish, we transfer it to the glass slide using a paintbrush. So D. C, D, B, A. Answer number 4. All right. Answer number 4. Good one. Which of the following is correct regarding polysaccharides? So be careful, right? They're asking the correct statement, right? They're asking the correct statement. Polysaccharides, they are large molecules but not macromolecules. Wrong, no? Polysaccharides are macromolecules, right? They have all have linear structure. No, there are so many branch polysaccharides. There are so many branch polysaccharides, right? So you can categorize polysaccharides based on what? Function, right? And what? Architecture, right? So those are the two classifications now. So architecture means what? You have linear and branch. Functional, you have so many, no, right? Structural polysaccharides, storage polysaccharides, like that. So they don't all have. There are so many branch polysaccharides as well. Hemicellulose has a linear structure formed from the polymerization of hexoses. What do you think? Hemicellulose, is it linear or branched? Hemicellulose, linear or branched? Hemicellulose, linear or branched? Yes, it's branched, right? Check your resource. These are direct MCQ points. Huh? These are direct theory paths that, I mean, there's nothing for you to understand, right? Hemicellulose is a branch polysaccharide, right? That's the theory. Glycogen is a structural polysaccharide in fungi. And now, can you see how they are trying to confuse you? Yes, glycogen is found in fungi, okay, but is it as structural? No, it's as storage. No, glycogen is a storage polysaccharide. It's how they store glucose. It's how they store glucose, right? So again, wrong. Some polysaccharides possess nitrogen. What is a nitrogen containing polysaccharide? What is a nitrogen containing polysaccharide? Anyone? I think very good, right? Chitin. Chitin is a nitrogen containing polysaccharide. So MCQ 3, answer number 5, right? Moving on, correct state, correct response regarding the cytoskeleton. Correct statement regarding cytoskeleton, right? They're asking the correct one. Contributes to maintain the shape of plant cells. Is it necessary? Is it necessary for plant cells to have a cytoskeleton? No, they have a much better supportive structure. Like if you take cytoskeleton, support and maintenance of cell shape is actually only part of the role played by the cytoskeleton. You understand? Cytoskeleton is actually a very, let's say, more physiologically active structure, right? A support is only a part of the role. So many other roles, right? What are the other things? Connect the intracellular environment with the extracellular components, right? Cell communication. So many roles are played by the cytoskeleton, addition to the support. But if you take the solely the maintenance of shape role, the cell wall is better. That is found in plant cells. But anyway, in animal cells, because they don't have a cell wall, they have the cytoskeleton to provide support. Microfilaments are composed of 3 intertwined strands of actin. Again, these are all theory, right? Is it 3? No. 2 strands of actin wrap around to form what? Okay, a microfilament, right? A microfilament. Microtubules are composed of 9 plus 2 columns of tubulin and right? They are trying to confuse you, right? Tubulin is the structural protein of microtubules. Yes, microtubules are composed of tubulin. But what is the arrangement of tubulin in microtubules? As 13 columns, right? Yes, 13 columns of tubulin microtubules are formed. Where do you have this 9 plus 2 arrangement? Where do you have this 9 plus 2 arrangement? That is found in where? In where? Why do you have this 9 plus 2 arrangement? Flagellum. Structure of what flagellum? Huh? What flagellum? So eukaryotes and prokaryotes both have flagella. Which flagellum? Whose flagellum? You have to be specific. Which? Whose flagellum? Whose flagellum? It's eukaryotic flagellum. Yes, I didn't disappear. Right? Microtubules are arranged in 9 plus 2 arrangement in the flagellum of eukaryotes. Okay. Eukaryotes. Right? Prokaryotes do not have a nice arrangement of flagella. Right? Yeah. There's another one. Intermediate filaments of all cells are made up of keratin. No. Keratin, I mean, intermediate filaments can be composed of a number of proteins. There's a number of proteins that can form intermediate filaments. Keratin is only one. තව තව ඒවායින් හැදෙන්න පුළුවන්. Intermediate filaments can be composed of many other things, right? Keratin is just one. Provides strength to the cytoplasm. Yes. Again, this MCQ, right? මේ ඔක්කොම දන්නේ නැති maths එක්කෙනෙක්ට දුන්නා, right? Cytoskeleton කියලා එකක් තියෙනවා. Maybe skeleton. අපේ skeleton එකෙන් what does it do? It provides some support, right? And so anyone with logic can ටක් ගාලා ගහන්න this fifth is the answer, right? Because it makes the most sense, right? මේ අනිත් if they don't know any of the MCQ, I mean, any of the theory points necessary, they can still get an answer, right? Now, be very careful. They're asking incorrect statement. Huh? They're asking incorrect statement. Be very, very careful. Most of the competitive inhibitors are non-reversible. Most of the competitive inhibitors, right? When you are doing these MCQs, highlight the keywords, right? Competitive inhibitors are non-reversible. Competitive means what? They are usually reversible. Usually competitive inhibition is reversible. How do you reverse a competitive inhibition? How do you reverse competitive inhibition? Then, for example, you are competing to get into medical faculty or something. You are competing with someone else. Who are you competing with? Another A-level candidate, right? Another A-level candidate you are competing with, right? Because in the exam situation, you and whatever the other A-level candidate is the same, right? You are given the same paper, you are writing it at a government exam hall at this almost the same conditions, right? So you two are equal. You are competing for what? For the same position in a medical faculty, right? So how can you compete with them, right? If you have a lot of competitors trying to get in, that is going to increase the blockage for someone else, right? When the number of candidates increase, the number of blocking, right? If you take a person trying to write the exam, A level, and trying to get into medicine from Gampaha district, which is the highest number of candidates, right? The competition is very high, right? So that sort of situation. So the only way that you can fight competitive inhibition is by increasing the number of substrates, right? So if you can fight an inhibition, that means what? It's a reversible one. No? So if it's non-reversible, can you fight it? No. ඒක set වුණොත් set වුණාම තමයි. You can't remove it. So they are not non-reversible. They are usually reversible. Molecules or ions are enzyme inhibitors. Molecules or ions are enzyme inhibitors. Yes, they can be enzyme inhibitors. Right? Molecules are what? Vitamins. Right? Can be. Sorry, inhibitors. No, they are asking inhibitors. Right? Certain you know ions such as heavy metals and molecules such as toxins and poisons can be in enzyme inhibitors, toxins and poisons, right? So that's okay. Drugs used against microbes are reversible inhibitors. Yes, these are again theory parts. Drugs used against microbes, particularly antibiotics. Antibiotics have reversible effects because why? We are using antibiotics within our body, no? All right, within our body, we are drinking or taking antibiotics IV directly into the blood system, right, into a vein, right? If they are irreversible, that means their toxicity is usually high, right? Usually in a irreversible inhibitor, the toxicity is high. So you can't usually use it in a living system like us, right? But you can use such sort of inhibitors, let's say in a bacteria culture, lab එකේ අපි culture කරන්නේ bacteria. You can put something very toxic like that into that sort of thing, but you can't take it into your body. That's that much something that is so much toxic. So usually they are reversible. So usually they are reversible. That is okay. Competitive inhibitors compete with the substrate for the active site. Yes, that is the definition. Right? Active site, be very careful of these key terms. Right? They are saying the active site. Yes, competitive inhibitors, they look like the substrate. They look like the substrate, so they compete for the active site. Non-competitive inhibitors change the shape of the active site. Yes, right? They join somewhere other than the active site, that is usually called the regulatory site, or you can say if it is under regulatory site, right? You can say if it is regulated by allosterism, you can say allosteric site, right? Whatever, it's a regulatory site. When they bind to the regulatory site, the shape of the active site will be changed, right? Answer number one. Answer number one. Question number 6. Intermediate compound formed in Calvin cycle as well as anaerobic respiration is ethanol. Calvin cycle එකේ ethanol හැදෙනවද? Right. Ethanol is a production in right ethyl alcohol fermentation. No. Right. Ethanol you don't have. Acetaldehyde. Acetaldehyde is only found in what? Acetaldehyde only found where? Yes, anaerobic respiration. What anaerobic respiration? Acetaldehyde is found in what respiration? As an intermediate product? Acetaldehyde. There is CO2 evolution. Ethyl alcohol again, right? Ethyl alcohol, right? If I remind you, what is ethyl alcohol fermentation look like? What is the starting product? Glucose, right? Glucose will be converted to what? Glucose will be converted to what? Glucose is how many carbons? C6 will be converted to yes, acetaldehyde. Acetaldehyde will be converted to what? Acetyl will be converted to what? Sorry, what's the first step? Glucose goes into pyruvate, pyruvate. So I gave you the answer. What is this process? What is this first process happening? That is common to any. That is common to any aerobic or anaerobic. Glycolysis. Glycolysis is happening. So pyruvate is how many carbons? Right? 3 carbon. Nah? This is 6 carbon glucose broken down into 2 pyruvates, which are 3 carbon each. Then the 2 pyruvates go into what? Acetaldehyde. In the process, what's happening? In the process, what's happening? Yes. Online people, can you hear me? Okay. Acetaldehyde has how many carbons? 2 carbons, no? So, what is happening in the middle? There is going to be evolution of carbon dioxide. Methane CO2 එකක් evolve වෙනවා, right? And we go on to the final step, that is ethyl alcohol, which is again C2H5OH, no? Right? C2H5OH. So, that is again 2 carbons, right? So, balance these numbers. There is 2 acetaldehydes and 2 ethyl alcohols produced, right? And this whole process, in this process, what is going to be happen? What is going to happen? NAD plus is converted to NADH. This NADH will convert back to NAD plus when converting the acetaldehyde back to, sorry, when acetaldehyde converts to ethyl alcohol, this NADH will convert back to NAD plus. Got it? Yep. So these are sort of the mind maps, right? Flow charts that you need to have in your mind. Okay. So using this knowledge, we can come back and I think we can do all the other questions, right? My annotations. Annotations are මොකද Where were we? Yeah. So ethanol is only found in ethyl alcohol, acetaldehyde, ethyl, G3P. Is there any G3P production in any of the anaerobic respirations? No. G3P only found in Calvin, which is used as what? The precursor, which is the precursor for, right? It's the precursor, no? For carbohydrates, precursor for usually for glucose. Malate, any malate production in anaerobic respiration? No. Not even in Calvin cycle, no? Right? Calvin cycle එකෙත් malate තියෙනවා. No. Pyruvate is right. Okay. Any questions? Right. Moving on to question number 7. Which of the following does not take place? Does not take place in the citric acid cycle. Citric acid cycle. So cycles, it's okay for you to be a little bit confused with all these cycles at this point, right? Citric acid cycle is a part of respiration, no? It's a part of respiration. So it does not take place in the citric acid cycle, right? CO2 is released from decarboxylation in citric acid cycle. Is there a CO2 release? Is there a CO2 release? What is the basic summary of citric acid cycle? What is the basic summary of citric acid cycle? Hmm? Huh? No, I can drag කරන්න පුළුවන්. I can slide it, right? Huh? හරි මම කියන්නේ it's infinite. You can sort of slide it. නැද්ද? No, hold on. Okay. Plus means new whiteboard. Okay, anyway, right? So we are in what? Citric acid cycle, right? So what do you feed into the citric acid cycle? What do you feed into the citric acid cycle? What is the starting product? Acetyl CoA. That is the feeding, no? That's what you feed into the citric acid cycle, right? Acetyl CoA combines with what? Oxaloacetate. Oxaloacetate. This combination produces what? Citric acid. So this is the starting part, no? Right? You have to be able to. How do you drag this? Yeah. Use the hand gesture mode. Where? Where do you have the hand gesture mode? Where do you have the hand gesture mode? But it's giving me only the erase. Why is that? Right cone? This one? Yeah, okay. So this circle. Where? Nope. This one. The other things? Which circle? Starting problems, no? Right? How do you. Anyway, right? So citric acid converts back to oxaloacetate. In the process, what's going to happen? Right? Citric acid is how many carbons? Acetyl CoA comes with 2 carbons. Oxaloacetate is 4 carbons. Citric acid is 6 carbons. All right. So in the process of citric acid becoming oxaloacetate, there's going to be 2 CO2 released. Right? 2 CO2 released. And what are the other products for one cycle? What are the other products for one cycle? Yes. What are the other products for supposed to redo? This one. It doesn't work. Right? I tried it. Only the eraser is correct. This one? Yeah, only the eraser is coming. Why is that? I'll try to figure it out. Okay. Right. So what are the other products? There's going to be one FADH2 and what else? Double click? What do you mean? I'm trying everything you say. Right? One FADH2 and three NADH produced. Right? Three NADH produced, and other than that, per cycle there's going to be one ATP. Usually it's we say it's GTP, right? But anyway, in your syllabus it says one ATP produced by what? Substrate level phosphorylation. Substrate level phosphorylation, right? Coming back to this is the PDF. Huh? Ah, yeah, nice, right? So where were we? Which of the following does not take place in citric acid cycle? CO2 is released. Yes. Regeneration of citrate from oxaloacetate. Are we regenerating citrate from oxaloacetate? No, right? Citrate dwelling, we are regenerating oxaloacetate. No, right? We are regenerating oxaloacetate from citrate, right? Other way around. So wrong. FADH2 is generated from oxidation. Is it oxidation? So this question, there was an issue, no? FADH2 is generated from oxidation. Yes. Because why? What do you initially have? You have FAD plus, right? FAD plus with when it gets electrons, it's going to become FADH2. All right. So this is what this is thing FADA plus is getting reduced, but what is the process happening for for citrate? Citrate is getting oxidized. Citrate is getting oxidized. Therefore, FAD plus is getting electrons. So it is undergoing reduction. Substrate level phosphorylation takes place. Yes, that is how we get the 1 ATP. Occur in the matrix of mitochondria. Is it occurring in the matrix of mitochondria? Yes. Citric acid cycle takes place in the matrix of mitochondria. Citric acid cycle takes place. Yes, yes. So, what is the confusion? Does not take place. It is generated from. I can understand how that can cause a bit of confusion. Statement number 3: FADH2 is generated from oxidation. So, usually we refer as the process. Citrate is getting oxidized. Citrate is getting oxidized. Statement number 2 is definitely wrong. As you can understand, it's the other way around. We are not generating citrate. It's not regenerating. No, we are regenerating oxaloacetate. Because why? We need oxaloacetate to do the other cycle, right? We need to do the cycle again and again. We need oxaloacetate, right? Yeah. FADH2 is generated by oxidation, right? Yes, it is getting reduced, but who is giving the electrons? That is mainly citrate. So the process that is triggering the FADH2 formation is an oxidation. That's the logic. Okay. Which of the following statement is correct regarding enzyme inhibitors? Again, we are coming to enzyme inhibitors, right? Certain molecules of ions bind to enzymes reversibly with covalent bonds. No, if it's reversible, if it's covalent bonds, it cannot be reversible. Effect of non-competitive inhibitors can be reversed by increasing the substrate concentration. No, we can't usually reverse the effect of non-competitive inhibitors, right? We have to give new enzymes, right? To remove the effect of non-competitive, the only thing we can do is new enzymes දෙන එක විතරයි. දැන් ඉන්න enzymes ටික they are beyond saving. අපිට ඒගොල්ලන්ව බේර ගන්න බෑ. They have been changed by the non-competitive inhibitors. right? So they cannot be reversed. Competitive inhibitors interrupt enzyme reaction by binding to the part of the enzyme other than the active site. No, competitive, they bind to the active site, right? Other than Neville, right? They bind to the active site. Drugs used against microbes can act as irreversible inhibitors. No, right? I told you they are usually less toxic, so they are usually reversible. Then non-competitive inhibitors decrease the efficiency of formation of enzyme substrate complex. Yes, because why? They change the shape of active site, right? So what do you need to form the enzyme substrate complex? You have to have the substrate, right? And the enzyme should have the active site with the proper what specificity. So they form a nice contact, right? Lock and key hypothesis. So when there is an uncompetitive inhibitor that is changing the shape of the active site, the enzyme substrate complex formation is affected. Okay. So answer number 5. Some events taking place in photosynthesis are listed below. Only happens during light dependent reaction. Only happens during light dependent reaction, right? So for this one, I think there was an issue. No, this question there was a small issue, right? No, number 9 issue එකක් තිබ්බනේ. Hold on, right? Let's see. Striking of photons of light, right? Only happens, yes, definitely during the light reaction, photons of light. I corrected this. I think all of you got this, right? I corrected this to photons. This is photons of light. Electrons. It has been auto corrected to electrons. Right? Autocorrect is Pandita Vedi Samarlavata. Right? It is photons of light. So definitely yes, in the light dependent reaction there is striking of photons. Yes. Reduction of NADP plus and FAD. No, definitely FADs. These things are related to what? Those are related to cellular respiration. Photosynthesis වලට ගෑවිලාවක්. Reduction of 3-PGA, that is again Calvin cycle. No, 3-PGA reduction happens in Calvin cycle. Synthesis of ATP by cyclic electron flow. Yes, right. Cyclic electron flow happens where? In the which photosystem? Cyclic electron flow happens in which photosystem? PS 1, right? Cyclic electron flow happens in photosystem 1, right? Electrons at the P700 goes through a cyclic pathway to come back to the primary electron acceptor, and by this process we get ATP, right? Only ATP, there is no NADH produced, right? There is no, sorry, NADPH produced, and it involves only photosystem 1, right? 7th question, answer number 2. Yes, does not take place. Yes, we take answer number 2. So this one, question number 9, PNS. During glycolysis of cellular respiration, glycolysis, synthesis of ATP and NADPH. There is no NADP, it's only NADH. Synthesis of two CO2 molecules by decarboxylation. There is no CO2 production in glycolysis, because why? How do we start glycolysis? We start glycolysis with glucose. We end up with two pyruvates. අපි පටන් ගන්න glucose එකකින් ඉවර වෙන්නේ pyruvate එකකින්. So glucose has how many carbons? 6 carbons. Pyruvate has 3 carbons. එතකොට carbon number එක It has not been changed. So has there been any CO2 evolution, any decarboxylation? No. Use molecule oxygen? No. Glycolysis is common to both aerobic and anaerobic. So there is no oxygen requirement. Reduction of coenzyme. So this is, we are meaning coenzyme A, yeah? Right? There is no reduction of coenzyme A happening in glycolysis. Where is reduction of coenzyme A happening? In the linking reaction. Linking reaction again. Glucose is reduced to pyruvates. Okay. Monomer of inulin is what? What is the name of the monomer first of all? What is the monomer of inulin first of all? Fructose, no? So then let's think about fructose. What are the things you know about fructose? Right? Is it a ketose? Sorry, is it an aldose? No, it's a ketose. Right? It's not an aldose, it's a ketose. Right? Fructose is a ketose. What are the two aldoses? What are the two aldoses? What are the two aldoses that you have under monosaccharides? Right? What are the two aldoses? Glucose and galactose. Glucose and galactose. So fructose is a ketone sugar, a component of sucrose. Sucrose, then do you need fructose? Do you need fructose to make sucrose? Yes. Glucose and fructose. It's a non-reducing sugar? No. All monosaccharides are reducing. All monosaccharides are reducing sugars. Is it a pentose? Fructose, is it a pentose? 5 carbon? No, these are all hexoses, no? Carbon hydroxide. These are all hexoses. 6 carbon monosaccharide. A hexose that is insoluble in water? No, right? What are the three properties of monosaccharides? Monosaccharide hexoses, water soluble, crystalline, sweet taste, and reducing, right? They are all water soluble. Answer number 2, no? Right. Question number 12. Which of the following statements about amino acids is correct? Every amino acid has an asymmetric carbon. Is it every? What is the exception? Are the amino acid who's having a symmetric carbon? Name me an amino acid. Amino acid name symmetric carbon. Glycine. Na? So here you have NH2. Here you have COOH. Udayatai. You have hydrogen. Asymmetric. This is what? This is symmetric. So glycine is an exception, so you can't take. Only R groups contribute to the formation of hydrogen bonds between amino acids in albumin protein. Albumin. What is albumin? Albumin, what is the structure? Level of protein structure. What is the level of protein structure? Why not answers, right? Right? It's a tertiary structure. For the formation of and the stabilization of tertiary structure, you need so many other bonds, right? And also we say intra and intermolecular, no? R groups and because in your secondary structure we have hydrogen bond formation between carboxyl and amino groups. So that is also involved up to the tertiary level. Tertiary level those hydrogen bonds are also there. So you can't say only R groups. You can't say only R groups. Only one carboxyl group can be present in any amino acid? No. There can be more than one carboxyl group. I can't remember the exact amino acid, right? So there's an amino acid whose R group is COOH, right? So they have two carboxyl groups, right? I can't exactly remember the amino acid, but there's an amino acid who's having COH as the R group. A peptide bond joins two amino acid molecules to form a dipeptide. Yes, amino acid දෙකක් set වුනහම two monopeptides to form a dipeptide. Peptide bond එකකින් join. The number of non-essential amino acids is 10. No. Right? What do you mean by non-essential? This is a bit out of your syllabus, not syllabus, but out of your level, right? It is actually coming in grade 12, lesson number 5, nutrition. All right. So, what are these essential and non-essential amino acids? Right. So, you know, what is the total number of amino acids that you need to make protein? 20. ඔක්කොම 20ක් ඕනේ. Right. So, මෙහෙම આવුවොත් the number of amino acids found in nature is 20. Total number of amino acids found in nature is 20. Is that true? No. There's only 20 amino acids contributing to make proteins. Proteins හදන්න 20ක් set වෙනවා, but there are more than 20 amino acids in nature. So that is one MCQ point. What is the other one? You can divide this 20, you can divide this 20 into two categories based on your body's ability to synthesize, right? You have, depending on your body's ability to synthesize. So there are things, there are amino acids that you definitely need to take in through your diet. Those are called as essential. Even though you cannot survive if you don't eat those amino acids, you are going to get a protein deficiency. How many essential ones are there? There's actually 8. මේ 8 අනිවාර්යයෙන්ම කන්න ඕනේ. Non-essential liver, there's 12, right? මේ 8 you can synthesize within your body. What is the main protein synthesizing organ in your body? What is the main protein synthesizing organ in your body? Liver. So your liver can synthesize these non-essential amino acids from these essential amino acids. මේ 8න් මේ 12 හදාගන්න පුළුවන්. Right? So usually we say plant protein products usually lack one or more essential amino acids. So what are your plant proteins that you know? Soya beans, germinating seeds, right? Green gram. What else? Usually plant products having protein usually lack at least one essential amino acid. That's why we say for a vegetarian, exclusive vegetarian, විතරවත් කන්න නැති vegetarian කෙනෙක්ට, it is essential that they include a variety of plant protein products. Right? They have to have a variety of plant protein products. For example, if a person eats eggs, that means if a person ate an egg, an egg is a super food. Right? You can consider an egg as a superfood because it contains all the essential amino acids and a lot of omega 3 and omega 6 and cholesterol necessary for hormone synthesis and cell repair and everything. Right? An egg is a superfood, but a person who's not eating even an egg definitely has to pay attention to their diet. They have to make sure that they are taking all the essential amino acids definitely. Right? Otherwise, they are going to have protein deficiencies. So, what's the answer? 3, no? For 12, answer number 3. No. Sorry, 4. Sorry, 4. Dipeptide katawa, no? Golgi apparatus is a stack of tubular sacs. It's a stack of tubular sacs. Right? Is it tubular? They are flattened, no? It's a stack of flattened sacs. Right? How do we draw Golgi? මෙහෙමනේ අඳින්නේ, right? Are they tubular? Right? මෙහෙමනේ අපි Golgi අඳින්නේ. Are they tubular? No, they are flattened. 5 eggs per day. That depends, right? That depends if you require the protein intake. So for the recommendation is at least one egg per day, right? That is for a normally active person. Depending on your requirement, if you are doing a lot of hypertrophy exercises, right? Weightlifting exercises focused on increasing muscle mass, then you definitely need to fuel the muscle, right? You need to give the building blocks, right? නිකන් ගෙයක් හදන්න බාසුන් ලැහැලා ගෙනල්ලා දාලා සිමෙන්ති වැලි දෙන්න නැතුව ඉන්න බෑ. එතකොට මොකද ඒගොල්ලෝ කරන්නේ? right? අල්ලපු ගෙවල් වලින් හොරකම් කරලා හරි ගේ හදන්න try කරනවා, right? So if you think of that example, so you are going to the gym. If you are going to the gym, that means what? You are trying to build a house. You are trying to build muscle, right? Gym එකට ගිහිල්ලා if you are not giving the building materials, that is the protein, then what is going to happen? Your muscles are going to get actually atrophied because you are asking too much of them. You are giving them too much stress, but you're not giving them the repair materials, right? So if you're not giving them the repair materials, the muscles are actually going to get weaker, atrophied, and sometimes so you are going to lose weight. You are going to lose muscle mass, right? So ඔයගොල්ලෝ බාසුන් හැල ගෙනලා දානවා නම් gym එකක් sorry මේ ගයක් හදන්න, you have to give the building materials, otherwise it's useless. But anyway, it's better to be careful of the cholesterol intake, right? Better to be careful of the cholesterol intake, but actually 5 eggs is not in a, I mean, it's not in a dangerous level of cholesterol even, right? Because in the age of 20 to 30, our testosterone levels are at a maximum. So testosterone is a steroid, no? Steroid, right? What is the precursor? What is a precursor for steroid hormones? What is a precursor for steroid hormones? What is the precursor for steroid hormones? Anyone? What is our starting material that we need to make any steroid product in our body? Steroid hormones mainly. Cholesterol. Right? It's a myth that cholesterol is bad. Right? Right? Mainly your what we call as dyslipidemia. Having අපි නිකන් cholesterol වැඩි වෙලා කියලා. Dyslipidemia is mainly genetical and also related to the lack of activity, lack of exercise, right? Lack of exercise. If you are a healthy person, it doesn't have to be a male, right? Even a female, you have steroid hormones, estrogen and progesterone. These are based on cholesterol. So you need cholesterol to be healthy, right? If you are active, right? If you're active and you don't go over, right? You don't go over the cholesterol intake, and particularly if you are taking healthy lipids, right? So cholesterol in your egg is a healthy lipid. What are the sort of lipids that are going to contribute to this dyslipidemia? LDL, low density lipoproteins, those are formed when you take unhealthy, right? What are the unhealthy fat containing, right? Foods? Refried. ආපහු refry කරපුවා, and also deep fried. මේ දෙකම ඉතින් very closely connected. Whatever the things that are deep fried are again refried in the same oil, right? So LDL is actually a vehicle that carries cholesterol, right? LDL is a vehicle that carries cholesterol. But if your body requires that cholesterol, if you are an active person that is going, I mean, you are making new cells and you are making a lot of hormones, that means that you are going to need that cholesterol, right? You are going to need that cholesterol. That means that cholesterol is not going to cause any harm, right? So there should be a proper balance, right? You have to be aware of what is your activity level, and if you have a genetic disposition, අම්මට තාත්තට පොඩ්ඩක් cholesterol තියෙනවා නම් it's good to be careful. But good cholesterols, particularly the egg cholesterols, are unlikely to cause any problems, right? Egg cholesterols, but do not go more than about 10 eggs per day. And if you are eating about 10 eggs, better to eat only about 5 egg yolks, අනිත් ඒවා egg whites කන්න, right? Better to take only about 5 whole eggs, right? Anyway, Question number 13. Na? Golgi apparatus is a stack of tubular sacs. So that is wrong. Produces vesicles containing hydrolytic enzymes. So what are they talking about here? What are they talking about here? What is your vesicle containing hydrolytic enzyme? It's not an eraser, right? Can you erase like a whole area? No. I'm still figuring this thing out. Right. So what is a vesicle containing hydrolytic enzymes? A lysosome. Alright. So do Golgi produce lysosomes? Do Golgi produce lysosomes? Golgi lysosome are the Yes. They do produce lysosomes. Produce vesicles from rough endoplasmic reticulum by its transphase. Right. So Golgi has two sides, no? There's a receiving side that is called what? Cis. The receiving side is called the cis, and the outgoing is called the trans. So this is the input side, cis phase. This is the output side, right? So they receive rough endoplasmic reticulum by its trans phase, no, by its cis phase. Involves in synthesizing, collecting, modifying, packaging, and distribution. Right? Make a kyoganyanti, it seems like all the things that are done by Golgi, but pay attention. Synthesizing, collecting, modifying, packaging, and distribution. Proteins and membrane lipids. Where is the problem? Anyone? Where is the problem? What is something not done by Golgi? Because the Golgi will කරන්නේ නැත්තේ. What is not done by Golgi? They don't synthesize proteins. They don't synthesize any proteins. They only collect, modify, package, and distribute. Huh? They don't synthesize. So yes, so that is wrong. Contributes to forming the cell plate by the vesicles formed by the cis phase. එතනත් ප්‍රශ්නයක් තියෙනවානේ. The vesicles are produced by what? Trans phase. නැත්නම් අනිත් ඔක්කොම okay. Yes, the cell plate is produced by the vesicles. That is good. But is it given out by the cis phase? No. Outgoing is what? Trans phase. Input cis. Outgoing trans. Which of the following is correct regarding enzymes? Which of the following is correct regarding enzymes? Toxins are often inhibitors that binds to the enzyme through weak bonds. No. Toxins bind do not bind to තමයි usual. Many competitive inhibitors are irreversible. No. Competitive now, usually reversible. Enzymes produce the energy difference between reactants and products. Reduce the energy difference between reactants and products. So if you take, this is the reaction. So these are the reactants. These are the products. Right? What is this gap? Sorry, not that big, no? So when you are doing chemical kinetics, have you started chemical kinetics? Not yet, right? So this is the energy jump. The reactants have to sort of, they have to go across that hurdle, right? They have to jump that hurdle. But what's the energy difference? This one, we call it delta H, right? Delta H kilokira, right? So that you will learn in chemical kinetics. So do they reduce the energy difference? No, right? So imagine we put an enzyme into this system. What is going to happen? The activation energy is going to be reduced. Only the activation energy is going to be reduced. This delta H is not going to be reduced. The energy difference is going to be the same, right? So enzymes only reduce the activation energy, not the energy difference. Delta H K it's going to be remain a constant, right? Vitamin B is a component of coenzyme NAD. I think that's the correct statement that we can take. No? Yes, vitamin B is a component of coenzyme NAD+. Binding of a substrate molecule to the regulatory site increases the catalytic activity of the enzyme, showing cooperativity. What's the problem? We are describing cooperativity here. We are describing cooperativity here. What happens in cooperativity? Yes, it's the binding of a substrate, but where? Go and check the resource book. It's to one of the active sites, right? Substrate the regulatory site bind, right? A substrate always binds to, yeah, active site. A regulatory molecule binds to regulatory site. Substrate binds to an active site. So in cooperativity, what happens? What's the best example that we have for cooperativity? Hemoglobin, no? Hemoglobin. When one oxygen binds to an active site, it increases the affinity of oxygen in all the other active sites. Anyone, any question? You understood, right? It's not binding to the regulatory site, it is binding to the active site. Huh? Got it? So answer number 4. Huh? Some events related to metabolic processes are given below. Question number 15. Is the paper difficult? Was the paper difficult? I was marking some MCQs. You have scored in the MCQ paper. No? MCQ paper scores are not bad, right? Particularly, I have to sort of discuss about your essay writing. That will I'll do once we are in our normal classes, right? Today I'll try to cover the MCQ and structured essay parts, right? Because it might be unfair for some people if we go through the essay discussion today as well, right? So let's finish MCQ and structured within today. Metabolic processes. What are the events that can be seen in the C4 pathway? C4. Regeneration of oxaloacetate by citric acid. Is it happening? Citric acid will oxaloacetate regenerate. No. So be careful. Nah? We are only asking online people marks. I didn't exactly analyze because I wasn't going to give marks today. I separately marked the MCQs and essays and structured. Right. Next week I'll analyze your marks. Huh? Right. Next week I'll analyze both physical and online people's marks. Huh? Okay. So be careful. We are only talking about the C4 pathway. Huh? The reactions that are exclusive to the C4. What is C4? It's a CO2 delivery mechanism. No, bundle sheet. Sorry, mesophyll එකේ ඉඳලා we are delivering CO2 to the bundle sheet. So think only about that, right? Only about the delivery pathway. So oxaloacetate regeneration, it's happening where? That's happening in citric acid cycle. No? Is it something that is found in C4? No. Malate to pyruvate. Malate to pyruvate conversion. Is it something happening in C4? Malate to pyruvate? Yes. No? Decarboxylation? Yes. Malate converts to pyruvate by releasing CO2. This CO2 will be accepted by who? RUBP. So this is the Calvin cycle taking place in the bundle sheath. So there's a carbon dioxide released by decarboxylation. Hydrolysis of ATP, yes. To take this pyruvate back to mesophyll cell, there's ATP. Then ATP code. Right? Go through my C4 pathway summary. Right? We drew the C4 pathway from the mesophyll to the bundle sheaths. Right? Regeneration of RUBP by G3P. This is not exactly C4, no? It's Kelvin side. The methane indala thiyenne, this is Kelvin. Right? That is not part of the C4 part. So what are the things you can say? B, C, D, no? Answer number 2. Yes, B, C, D. Any doubts? Yeah, there are people who can't join the session today. Yeah, that's a bit unfortunate. They are having a power cut. Yeah, that's unfortunate. Right? So, yeah, let's quickly conclude the MCQ paper. 15, okay, no? Right? Regarding cellular respiration is correct. Pyruvate molecules produced during glycolysis enter the mitochondria passively through the membrane. Let's keep it right. In the citric acid cycle, 4 CO2 molecules are released for each glucose molecule. Yes, no? You have citric acid, which is 6 carbon. You have oxaloacetate, which is 4 carbon, right? This is 6 carbon. So per one cycle of the citric acid cycle, you release 2 CO2. But from one glucose, how many citric acid cycles you get? From one glucose, you can run the citric acid cycle 2 times. So you get per glucose 4 carbon dioxides released. Understood, right? Any doubts about that? Per glucose, 4 CO2s are released, right? 2 ATP molecules are used to transport 2 NADH molecules from the matrix to crystal. Mitochondria එක ඇතුලේ transport කරන්න you don't need any ATP. Respiratory quotient of protein is 0.7. No. What is it? What is the respiratory quotient of protein? These are direct MCQ points. It's 0.8, not 0.7. Nah? 0.7 is for what, Puta? Fats. Fats. Huh? Glycerol enters the citric acid cycle as acetyl CoA. No, glycerol enters the glycolysis. No? Glycerol enters glycolysis, right? You have in your resource book, second lesson, last page. Second lesson, last page. Yes, can you see this page? The very last one. When you break down lipids, you get fatty acids and glycerol. Glycerol enter glycolysis as glyceraldehyde. G3P විදියට it enters, not acetyl CoA. Acetyl CoA විදියට එන්නේ who? Fatty acids. Okay. Hmm. 17. Which of the following not essential for an individual organism? Imagine you are surviving in the wild, right? කැලේ අතරමං වෙලා ඉන්නවා. You are an individual organism. What do you need to survive? What do you need to survive? Yes, you need to adapt to the environmental conditions. Do you need to reproduce? Maybe for your mental health, right? But do you need to reproduce to survive? For your own survival? As an individual organism, no. You need to metabolize, you need to produce energy, you need to repair your body, you need to metabolize. You need order and organization, right? You need to have a proper organization of your cells, tissues, organ systems. You need to have irritability and coordination. You need to have stimuli coming in. You need to be able to coordinate your muscles, skeletals, nerves, and everything in order to survive. You don't need to reproduce to survive as an individual organism. Very, very important. Individual. As a population, if you cannot reproduce, then it's a problem. As a population, it is essential to reproduce. Human population, it is essential to reproduce for the continuity of the species. But an individual organism does not need to reproduce to survive. Property of water acting as a thermal buffer. I'm going to go a bit faster, right? Acting as a thermal buffer, obviously, what? Ability to moderate temperature, right? Right? Ability to moderate temperature, obviously, right? Very, very easy question. Correct statement regarding carbohydrates. Galactose is a hexosis, but is it keto? No, it's a aldehyde. Hydrogen to oxygen ratio is higher than 2 to 1 in disaccharides? No, right? You can check C6, H12, O6. Now, right? Produces a disaccharide. What is released in the process? One water molecule is released. So, what is the equation of a disaccharide? C12, H22, O, how much? O11. 2 to 1, no? So, in a disaccharide, the ratio is different, no? Same as 2 to 1. Hemicellulose is a branch structural, right? Same thing repeated again, nah? Right? So, now we are asking, we are saying it's branch and structural, right? That is a correct statement. Inulin is a stored polysaccharide made of fructose. For 19 is the problem, no? Methane disaccharide. Very sorry, yeah. Right, it says disaccharide, right? Hemicellulose is a disaccharide. Right? Hemicellulose is a polysaccharide. So, statement number 3 is wrong. Inulin is a stored polysaccharide made up of fructose. Yes. Arthropoda cell wall has chitin. Okay. Arthropods, they have chitin, but where? Where do they have chitin? Arthropods, where do they have chitin? Do they have a cell wall? Animals, arthropods, insects, do they have cell walls? No. They have what we call as an external skeleton or an exoskeleton, right? Outside skeleton. So that is composed of what? Chitin. Have you ever මැරිච්ච කැරපොත්තෙක් පාගලා තියෙනවද? Dead cockroach crispy, no? Right? There's a sort of a very crispy sound if you rat a kalure, if you're going to the bathroom, there's a dead cockroach, right? That's because why? Once the inner organs of the cockroach has been decomposed, දිරලා ගියාට පස්සේ the chitinous exoskeleton will remain because it's sort of resistant. අපේ skeleton එක remain. What sort of skeleton do we have? We have ඇතුලේ skeleton, endoskeleton. Arthropods have an outer one, exoskeleton. Correct about organic compounds. So we come to right. Correct about organic compounds. Question number 20. Yeah, this one was the issue, no? Make the issue active. This question I gave all. This question I gave all. Saturated fats are made up of fatty acids with hydrocarbon chains having no double bonds. Yes. Right. Okay. No. Right. There are no double bonds. Double bonds tibbot මොකද වෙන්නේ? It will bend. Right. Double bonds tibbot it will bend. That means it won't pack nicely. It won't pack nicely. That means what? They usually exist in liquid state. Right. All amino acids have asymmetric carbon. Again, we are repeating. Glycine does not have a asymmetric carbon. Secondary structure of protein is formed as a result of bonds between H atom of the carboxyl and N atom of amino. Yes. Right. NH2 group එකේ මෙම hydrogens, you have two hydrogens and the COOH has oxygen. So these hydrogens and oxygens form the hydrogen bonds. Pyrimidine N has a single ring. Yes, pyrimidines are smaller. Pyrimidines are the smaller bases. Right. Purines are two rings, bigger ones. So this one, so A, C, D. There's another one. NAD acts as an oxidative agent. Oxidative. Oxidative means what? He's making someone else oxidize. Yeah, oxidative කරන. Again, oxidate is making someone else become oxidized. Yes. What do you think? So NAD, what can happen to NAD? NAD plus accepts electrons and become NADH. So that means he is getting reduced. If he is getting reduced, someone is getting oxidized. So he is an oxidative agent. Yes. So because of this, there's no proper answer. So I gave all. It's an all. So C is wrong. Why wrong? No, it is. It says hydrogen atom of carboxyl and N atom of amino. No. Yeah. N atom එක සම්බන්ධ වෙන්නේ. Right. The N atom, H atom of carboxyl, N atom of amino. No. Yes. Right. It's the hydrogen of the amino and oxygen of the carboxyl. So anyway, still the answers are A, D, E. No. Yeah. ADE. Right. ADE. So there's no answer. Right. ADE. There's no combination with ADE. Okay. So that's the minimum amount of marks you can get. Anyone ඕනම කෙනෙක්ට at least 1 out of 24 ගන්න පුළුවන්. Right. Coming to the final set of questions. Right. Question number 21. Correct statement regarding water. These are easy questions. Right. Water is at liquid form. When water is at liquid form, hydrogen bonds form, break, and reform in low frequency. No. Liquid now, it is breaking and reforming in very high frequency. Right. Charge of the oxygen atom in water molecules is slightly positive, while the H atom is slightly negative. No, these are nonsense. No. In a water molecule, there's an uneven distribution of charge between hydrogen and oxygen. Yes. If you draw a water molecule, because of what? Electronegativity difference. Because of the electronegativity difference, hydrogens are slightly positive, oxygen is slightly negative, because the oxygen is very electronegative. Okay. Due to adhesive forces of water molecules, water scatters. Is it adhesion, Puta? No. Adhesion means what? Water and other molecules, right? But water scatters walk on surface because of cohesive, no? Cohesion, right? Water attraction, water molecules. That is what makes the surface tension. Due to high specific heat capacity, an organism can release a large amount of heat with the minimum loss of water. This is due to what property? Evaporation, no? High heat of evaporation or vaporization in the resource, right? Obviously, when on a liquid surface, when the liquid water, right, H2O L changes its state to H2O gas, there's a energy release. That is actually latent heat of evaporation. No? You will do this. That is called the latent heat. No, latent heat. Right. You will do these things in physics. Right. You need something called a latent heat for the state change. Liquid in the gas when you need to. There's a energy change. Right. So that energy change is what is going to cool our body. That energy change is going. It's what's going to cool our body. So answer 5 didn't make it, right? There is no proper combination. Only C is correct, right? There is no proper combination. Correct statement regarding enzymes. Gain enzymes. කවුද මන්ද මේක හැදුවේ? Right. Uh, enzyme has regulatory site where the enzymatic reaction happens. No, regulatory site එක නෙවෙයි වෙන්නේ. Active site. Okay. All enzymes are globular proteins. Statement number B. What do you think? Yes. Are all enzymes globular proteins? Okay, give me the what is the exception? Give me another. Okay, proteins. What else can be enzymes? What else can be enzymes? Name another type of molecule that act as enzymes other than proteins. You are close. Be more tachuttak precise in tachuttak. What? Okay, RNA is okay. What kind of RNA? rRNA, no? Ribosomal RNA. Ribosomal RNA. Because in the ribosome, there is an enzymatic activity done by the ribosomal RNA that is actually a component of the ribosome. rRNA, very good, right? Even if you remember RNA can act as enzymes, that's good. Optimum pH for trypsin. Trypsin is a small intestinal enzyme. Small intestinal enzyme, trypsin, right? Usually in the small intestine, we have bicarbonate ions. Bicarbonate, HCO3 minus, released by your pancreas. Pancreas क्या नाम का? Agnash. Right? Pancreas. So that is going to make the pH of the small intestine basic, more towards the basic side. So सामान्य pH 7 onwards up to pH 14 is our basic side. Basic right? So in the small intestine, the pH is about 8. So trypsin is a small intestinal enzyme. All right. Drugs used again, microbes are under reversible inhibitors. Yes, this is true. We had the same statement again and again. ADP stimulates the ATP production in anabolism. What do you think? ADP and ATP. What do you need to make ADP? What do you need to convert ADP to ATP? ATP is having higher energy. So what do you need to do? You need to provide energy. Negative, you have to give energy. No? You have to, if you want to make ATP from ADP, you have to give energy. So, what sort of reactions release energy? Is it anabolism? No. Catabolism. You have to break down something to release energy. That energy will be used to convert ADP to ATP. Right? So that is what's wrong about this. ADP stimulates ATP production in catabolism. Catabolism. So what are the statements? C and D. So answer number 4. Compounds found in the bundle sheath cells of C4. I corrected this. This is C4, right? Of C4 plants. What do you have in bundle sheaths? Hmm? What do you have? Oxaloacetate. Do you have? Yes. Where? Mm. So in this question, you have to think about the cell as a whole, right? That whole cell is doing cellular respiration. It is part of the C4 pathway, right? That cell is doing cellular respiration as well, other than photosynthesis. එතකොට ඒවත් හිතන්න. All right. Malate. Do you have malate in the bundle sheath cells? What do you think? Do you have malate in the bundle sheath cells? So, pep, um, so phosphoenol pyruvate found in bundle sheath cells. What do you think? Bit doubtful, no? Because PEP is actually converted to oxaloacetate in the mesophyll cells. So we don't find PEP anywhere in else in the bundle sheath, no? So you can't take PEP. What do you think? Can you take PEP? Oxaloacetate you can. Because why? Oxaloacetate is part of this oxaloacetate. You can say it's part of citric acid cycle like that. Malate definitely. Malate ne අපිට bundle mesophyll එකේ ඉඳලා එන්නේ malate no, right? So malate definitely you can take. Pyruvate, yes, pyruvate is part of again you can say glycolysis and linking reaction, right? And this is the important one. P680. Do you have P680 in bundle sheath cells? What is the interesting thing about P680? It's the place where water is broken down and oxygen is released. P680 වලනේ oxygen release එක වෙන්නේ. So in the bundle sheath cell, do we try to remove oxygen or do we, what do we try to do? We always try to keep oxygen away from bundle sheath. Because why? Because why? If oxygen comes anywhere near the rubisco, what is going to happen? Photorespiration. Photorespiration. So in the chloroplasts found in bundle sheath cells, usually what? Thylakoids and grana are actually very reduced. Sometimes grana can be absent. Even if you have grana, they don't have photosystem 1. They don't have photosystem 1 means what? They don't have P680, right? So definitely E is wrong. Okay, definitely E is wrong. Then BCD, no, BCD. We have to take. What do you think? Answer number 5, no? Yes. Any questions? Yes, yes. So there's a question, right? There's a question. So formation of ATP itself is an anabolic reaction, තමයි, right? දැන් අපි දැන් මට මේකට අලුත් බෝඩ් එකක් ගන්න වෙනවා දැන් වෙලා, right? Right. Until I figure this thing out. Okay. So ADP plus inorganic phosphate becomes ATP. So which is the more high energy side? This one. Energy rich molecule, right? So yes, you have to provide energy. This reaction absorbs energy. So this is actually an anabolic reaction, yes. Make anabolic reaction එකක් තමයි. But who is giving this energy for this anabolic reaction? Who is giving this energy? It's by another reaction that is catabolic, right? This reaction is anabolic, yes. Someone else is undergoing a catabolic reaction in order to give the energy for this, right? In order to give the energy for this, right? Someone has to release energy for this one to take energy, right? Got it? Okay. Next question. Last one. Correct statement regarding photosynthesis. Major limiting factor is light intensity. No, usually in the normal environment, it is what? CO2 concentration, that is the limiting factor. Maize has high carboxylation efficiency than wheat. Maize is C4. Maize is C4, therefore the efficiency is usually high. Violet has less efficiency in simulating photosynthesis than green. No. Green වලට වඩා මොන පාට වුණත් efficient. Right? Green is the least efficient. So anything is more efficient than green. Any other color. Any other color is more efficient than green. NADP reductase is used in cyclic electron flow. No. I talked about cyclic electron flow earlier. Right? NADP reductase, that's only found in linear electron flow. Right? ලස්සන්ට electrons ටික from water ඉඳලා NADP plus වෙනකන් යන්නේ, that is in the linear electron flow. Cyclic electron flow, only around the photosystem 1, only ATP is produced. There is no NADPH product. High efficiency of the C4, this is C4 puta again, right? C4 photosynthesis is caused by the usage of less types of enzymes. Usage of less types of enzymes. Is it by the less types of enzymes? No. Less amount of enzymes. No? Less amount of enzymes. But I mean, think logically. If you take a look at the C4 pathway, it actually has more enzymes, right? There's pep carboxylase and everything, right? So the C4 pathway actually has more types of enzymes, but the efficiency comes with what? Less amount of enzymes. So you actually need less amount of Rubisco. The amount of Rubisco necessary is actually less. Therefore, the C4 pathway is efficient. Why? It leads to better nitrogen use efficiency. I told you nitrogen is one of the most The red, let's say least available nutrients in the soil. Nitrogen is one of the least available nutrients in the soil. So plants really need to manage their nitrogen. Plants really need to manage their nitrogen. And if they can manage without a lot of nitrogen, that is good. That means nitrogen deficiencies are not going to be a problem for that sort of plants. Got it? So what's the answer? Again, answer number 5, no? Make a wrong, make a okay. Violet take a wrong. Yeah, only B is correct. No? Yeah, answer number 5. Structured essay. Structured essay. Let's quickly discuss. I'll be giving the answers, right? I'll be giving the answers. What type of prokaryotes first form on Earth? Heterotrophic, anaerobic. Huh? Heterotrophic, anaerobic. Right? There was no oxygen, so they had to be anaerobic. There is no photosynthesis. They were heterotrophic. Right? Reproduction is a characteristic of organisms. Define it. Ability to produce offsprings for the continued existence of species. Right? So the keywords are offspring and continued existence. Continued existence of very important word species. අපි reproduce කරන්නේ අපේ නිකන් benefit එකකටවත් for the fun of it not, right? It's not for the fun of it that we reproduce. It is for the future of the species that we reproduce. Okay. Name two nucleosides containing bases belonging to the purines. What are the two purines that you have? Adenine, guanine. So adenosine and guanosine. Places where mRNA can be present in a cell. In a cell, so you can say nucleus. So from the nucleus, where do they go? Through the nucleopores into ribosome. So those are the two places where you can have mRNA. Or instead of ribosome, if you have written site of protein synthesis, it's also okay. Respiratory pigment of vertebrate muscle, puta, right? Muscle, better answer is myoglobin, right? Better answer is myoglobin, but I have given for hemoglobin as well, right? Hemoglobin and myoglobin. Can you hear me? Can you hear me? There's an announcement happening on the road. My voice is clear, right? My voice is clear, right? Okay, okay. There's an announcement vehicle going on the road, right? So hemoglobin, it's quaternary, right? Hemoglobin is quaternary, but if you have written myoglobin, it's tertiary. Yeah? Myoglobin is tertiary. Matrix of bone. What is the protein? They are asking protein. Nah? What is the protein found in bone? It's collagen. Collagen is quaternary structure. Okay, collagen is quaternary structure. Reagents that can be used to show that egg white contains protein. What is the test that we are going to do? Biuret. We are going to do biuret test. Right. What are the reagents? You can use either KOH or NaOH and copper sulfate. These are the reagents. Type of cell junction found in skin epithelium. Tight junction. Function is what? Prevent leakage of extracellular fluids into the intracellular spaces, right? Humming. I always say when you are swimming in a swimming pool, you don't get filled with water, no? That's because your skin has tight junctions. What is the function of fibronectin, right? Today itself, revise extracellular matrix. If you can remember, if this is a protein, right, and these are your phospholipids now, these are your phospholipids. There's a small protein piece here that is connecting your inner cytoskeletal fibers, cytoskeletal elements to the protein. This green color one is called fibronectin, right? So we can say it connects cytoskeleton to the integral proteins of plasma membrane. You can say this is what? This is an integral protein. You can say it connects the cytoskeleton to the integral proteins, or you can say involved in what? Communication කියලා කියන්න පුළුවන් නේද? I had another answer for this. What does it say under ECM? Hold on. You can say fibronectin. Yeah, the best answer is collagen fibers are connected to the integral proteins of plasma membrane. That is the function of fibronectin. That is the function of fibronectin. Collagen fibers are connected to the integral proteins. You can say it can. You can say it links extracellular matrix and cytoskeleton. You can also say that, right? It links extracellular matrix and cytoskeleton. You can say that as well. Okay. What is known as metastasis? What is known as metastasis? Spread of cancer cells to locations distant from their original location. So the keywords, right? Distant අනිවාර්යෙන් වෙන්න ඕනේ, right? And far distant from their original location, right? You have to also say I think cancer cells කියන එක ඉතින් නැත්තම් ඉතින් obviously no marks, right? How do tumor cells travel to other parts of the body, right? අපිට cancer cells වලට තියෙන highways මොනවාද in your body? What are the highways that your cancer cells can take? Blood, circulatory system, you can say blood vessels, and there's another sort of a transport system in our body that you will learn under circulatory systems in animals, that is called the lymphatic system, right? Lymphatic system. Think of the lymphatic system as sort of like a drainage system. That's how I usually explain, right? So your blood circulation usually cannot, so a blood circulation brings blood to cells and it also takes blood away from cells. But sometimes there is some amount of fluid remaining around cells, around tissues, that is called as tissue fluid. Sometimes this tissue fluid is too much, right? So usually your river does what? River brings water and also it takes away the water. In certain conditions, sometimes the amount of water is too much. So you need a sort of a flood control. Canal systems. So the lymphatic system is like that. It is there to prevent the accumulation of too much tissue fluid, right? So through this lymphatic system also, you can, the cancers can spread or they can metastasize. Name two plant growth regulators if not maintaining proper balance, post goals, auxin and cytokinin. Auxin and cytokinin. pH is a factor affecting the rate of enzymatic reactions. Why does enzymatic activity decline when pH is lower or higher? So what are we asking to describe? Optimum pH, right? So what happens when pH goes above or below? Alteration of chemical bonds involved in formation of enzyme substrate complex, right? There is going to be alteration. You have to say alteration is a keyword, right? What do I keyword කියලා කියන්නේ? If you miss one of these keywords, you're going to lose mark, right? But after a certain time, if you lose most of the keywords, more keywords ඕන ටික ඔක්කොම වගේ නැත්නම් zero marks, right? So alteration of chemical bonds involved in formation of enzyme substrate complex. Mention the modifications in the leaf architecture of C4 plants for more efficient photosynthesis compared to C3. So what is the modification? Right. So you have to, what you have to describe? You have to describe Kran's anatomy. You have to say, right? No need to, I mean, Kran's anatomy. If you no need to write Kran's anatomy, what are the components of Kran's anatomy? Bundle sheath cells. You have to say bundle sheath cells are enlarged and they have a higher organelle content, right? Bundle sheath cells are enlarged and they have a higher organelle content, and also, and what are they asking? They are asking efficient. Huh? So how do we make the C4 pathway more efficient? That is by we connect mesophyll and bundle sheath cells tightly interconnected to each other by plasmodesmata. Those are the 2 points, right? So those are the two components uh, of the C4. Let's say the Kran's anatomy that you have to mention here. State the type of cell in which the following events occur in the C4 pathway. These are again, and again this one, there's another point that you need to add. Bundle sheath cells enclose the vascular bundles, right? So these are the vascular bundles. These are the bundle sheath cells, right? And themselves are enclosed by what? Bundle sheath cells are enclosed by what? Bundle sheath cells themselves are surrounded by mesophyll cells. So there are three things you have to write here. Bundle sheath cells are enlarged and have a higher organelle content. Bundle sheath cells and mesophyll cells are tightly interconnected by a high number of plasmodesmata. And bundle sheath cells enclose the vascular bundles and are themselves surrounded by mesophyll cells. Those are the points that you have to mention in this question. Right. State the type of cells in which of the following events occur in the C4 pathway. These are direct MCQ points, right? Malate becoming pyruvate. Malate becomes pyruvate, releasing CO2. That happens where? In bundle sheath. So enol pyruvate regeneration that happens in mesophyll, no? Right? If regeneration happens in mesophyll. Formation of 3-PGA, this is part of what? This is part of Calvin cycle. No? So Calvin cycle takes place where? Bundle sheaths. Okay. Formation of 3-PGA is part of Calvin cycle. So that happens in bundle sheath cells. So that is the end of structured essay number 1. Any question? Any question? Am I going too fast? Right? Essays we'll discuss next week, right? So for this question, for your information, there's 40 points. I gave multiplied by 2.5 to give you total of 100 points, right? If you have gotten 40 out of 40 points, you are going to get 100 points. Okay, that was the marking scheme. What is the structural difference between a nucleotide and a nucleoside? right? Again, bit of repetition. A nucleotide without a phosphate group is called a nucleoside, right? There is no phosphate group. Phosphate group එක නැති වුණා කියන්නේ it's a nucleoside. Name two ions present in plant cell sap. Where do you have the cell sap? Where do you have the cell sap? Ismata, where do you have the cell sap? Yes, in the vacuoles. Okay. So, what are the ions? K plus, potassium ඉන්නවනේ, chloride ඉන්නවා. At which phase of mitosis nuclear envelope fragmented? I told you to be very very careful about this phase particularly. මේ nuclear envelope එක fragment වෙන්නේ කොතනදද කියන එක. It's a very very commonly asked question. It's not. It's sorry. It's pro meta phase. පුතා? It's pro meta එකේ තමයි fragmentation එක වෙන්නේ. State the specific site of occurrence of each of the following in a mitochondrion. In a mitochondrion, oxidative phosphorylation. ඒ කියන්නේ අපේ high energy containing molecules NADH and FADH2, they release electrons, and that electrons will be used to produce, used to create a storage of protons, and those protons are going to power ATP synthase enzyme, right? So whatever oxidative phosphorylation, that sort of are නිකන් dam එකකින් what are we doing? We are harvesting energy from stored protons, no? H plus ions ගොඩ ගහලා ඒ වගේන් අපි ATP හදනවා, right? Refer the notes, right? So that happens where you have to say very very clearly it takes place across cristae, right, of a mitochondrion කියලා ලිව්වෙන්නේ නැත්තත් okay, because we are asking given in a mitochondrion කියලා දීලා තියෙන හින්දා. But better in your A level exam, don't take a risk. Across cristae of mitochondria කියලා ලියන්න. Across the cristae of mitochondria. Conversion of pyruvate to acetyl. Pyruvate එකක් acetyl group එකකට convert කරන්නේ කොහෙද? That is in the linking reaction, no? So linking reaction takes place where? After the pyruvates have come into the mitochondria. So you have to say in the matrix of mitochondria. What is known as respiratory quotient, right? It is, you have to write down, it's the ratio of CO2 evolved and oxygen absorbed. Is the ratio of CO2 evolved and oxygen absorbed. For in a given time, right, you have to write these things. Given time කියන එක නැත්තම් you are going to lose mark for a what? Respiratory substrate. Right, it's a ratio between CO2 evolved and oxygen absorbed in a given time for a respiratory substrate. Following are some of the characteristics seen in living organisms. Explain what is meant by each of the following characteristics. So we are directly කෙලින්ම gediya පිටින් we are asking the definition. Growth is irreversible increase in dry mass, right? Right, what are the keywords? Irreversible, dry mass. Development, irreversible changes that takes place in the lifespan of an organism, right? Irreversible changes. Okay. Reproduction, ability of a ability to produce offsprings for the continuous existence. I asked this Isellath also, right? Of a species, puta, right? Very very important, mention the word species. Okay. 4 main types of organic compounds found in organisms. What is the main type in each of the following? Egg white. Kaleking, I didn't ask an answer. What is the main Organic compound found in egg whites. Yes. Sleepy. No. Protein. No. Coconut milk. Coconut milk. පොල් කිරි වලින් නේද මොනවද ගන්නෙ? පොල් තෙල්. right? පොල් තෙල් ගන්නෙ පොල් කිරි වලින්. Lipids. Primary cell walls mainly composed of cellulose and hemicellulose. Those are carbs. Arthropod exoskeleton, chitin, carb, carbohydrate. Name a laboratory test used to identify the following organic compound found in egg white. Again, I'm repeating myself, right? This is biuret. So I think it's the paper is easy, no? Structure essay paper is somewhat easy, no? What do you think? No, the word species is important for the definition of reproduction. Okay, the word species is important. Okay. Main organic compound found in coconut milk. What do you use to do to identify lipids? Any new student who didn't join with our practical sessions? Anyone hands up who didn't join with the practical sessions for the identification of organic compounds? Okay, I'll send. Those are available in my YouTube channel, right, as a public playlist, right? This is the YouTube channel. Where is it? Right. Where? Yeah. Scan this, and you will be directed to the YouTube channel. There you have a public playlist, practical seminar. Okay, there you will have all these identification of organic compound practicals. Okay. MCQs. I'll come to the marking schemes. Okay. Okay. State the common characteristic of all monosaccharides and some disaccharides. So this is a bit of a tricky place, right? We are asking the common characteristic of all monosaccharides and some. The answer I'm expecting is reducing ability. Reducing ability කියන්න පුළුවන් or reducing sugar කියලා කියන්න පුළුවන්. Oh yeah, but if you have written water soluble or crystalline and those things I have also given, but the because I can understand it's a bit confusing, but the answer I was expecting is this, because what I'm asking නිකන් some කියපු ගාම ඕගලන්ට තේරෙන්න ඕනේ I'm trying to නිකන් sucrose පොඩ්ඩක් we are trying to ignore the sucrose a little bit, right? So reducing ability is the answer I was expecting, but I have given to water solubility, crystalline nature, all those things I have given. Describe a simple lab test to identify sugars with the characteristic. So methanadi, now you should have understood what I am asking here, right? What the characteristic stated above. So what is the test that we do? It is a Benedict test, no? So you have to describe the Benedict's test. How do we do the Benedict's test? We add a sugar solution. We say usually 2%. 2% of the sugar solution into a test tube, right? May percentage එක නැති වුණා කියලා I mean it's not a keyword, right? 2% sugar solution into a test tube. Then we add equal volume. Very very important. මේක නැත්තම් ලකුණක් අඩු වෙනවා. Equal volume of Benedict. You have to add equal volume of Benedict reagent. Then you have to heat until boils. You have to heat until boils. Then I have given marks. What is the initial color? Blue. Red precipitate එනවා කියන එකට I have given marks, right? Describe the simple lab test. Final question. How is ATP synthesizing enzyme allosterically regulated? Allosterically regulated. So, allosteric regulation of ATP synthase, right? What are the things you need to know? You have to say ADP is an allosteric activator. ADP is an allosteric activator. It binds to the enzyme and stimulates. Okay, so ADP stimulates. You know this, right? ADP stimulates. Okay, stimulates the ATP production by catabolism. Also, you have to say by catabolism, because catabolism provides the energy. If the supply of ATP exceeds demand, then next point, if ATP is too much, if ATP is too much, the catabolism slows down and ATP act as inhibitor. So ATP itself act as inhibitor to the ATP synthase enzyme. So ADP is activator, ATP is inhibitor. You have to know, write those two things, and also you have to say what is the type of yeah, it is given allosterically regulated. So for this question, there's 34 maximum points, but for any 33 multiplied by 3 is going to give you 100 marks, right? Right. So that is the paper. Send your answers to the main chapter. Any questions that you have? Any questions? Essays I will discuss in the next class, right? Because I have to sort of walk you through how to write an essay properly. There's a bit of a confusion I can understand while I'm going through your marking schemes. I figured it's better if I can walk you through how to write essays using these two questions, particularly this one, right? This one, the second essay is actually pretty straightforward. Samar Lama a bit confused about how to write this sort of a long essay, aerobic respiration. So in that case, I'll be discussing how many marks for MCQs. So there's a 24. No? So 24. If you have 24 out of 24, it's going to be the maximum, right? So multiplied by 2, it's going to give you marks out of 50 by 50, right? That's the logic. Okay, right? MCQ paper half marks, and okay, for the two essay papers 15 neva dekai, and no arakata structured essay dekata 10 gane dekai ne. Yeah, I mean, I'm going to 150, I'm going to narrow it down to 15. Okay, I need to figure out the smart board as. So for the structured essay 100, I'm going to narrow it down to 10. Na? See the height again, no? 150, I'm going to take it to 15. Multiply by 2, then the total I will give out of 100. Got it? Okay. So with that, I think we can conclude for the day. Any question that you have? Any question? first in structured essay. Which one? Please send your questions to the main chat. It's difficult for me to keep opening individual chats. I think අපි කීර මේකට කියලා දුන්නා. Multiplied by 2.5, no? Right. 40 multiplied by 2.5 because it was a bit shorter. Sorry, longer, no? Longer question. Question number 1 was a bit longer. Okay, so we can conclude for the day, right? People who didn't join, you can watch the recording, and everyone who also joined, I hope if you're having any trouble with the weather, everything will be okay, right? So get back to your normal routine, right? By tomorrow, study, go through the discussion. If you have any doubts, you can ask in the next class, right? Yeah, so there's no pressure for you yet, right? There's no pressure for you yet. This paper was to give you just a taste, right? What taste එකක් දෙන්න, how to tackle MCQs, how to write structured essay answers, what do you mean by a keyword, right? How to write essays, right? How to manage your time, right? Even if you are done early, how can you use the remaining time to gather some more marks, right? ඔයගලන්ට 3 hours වලින් 2 hours වලින් paper එක ඉවරුණා කියලා you are not allowed to go out, right? So even if you are done within the remaining time, how do you gather more marks, right? So that is a skill you have to develop with time, right? So memorization as well as how to recall and how to how to input, how to convert your memory into the written answer is a art that comes with practice. Just because you have a good memory, sometimes it's not going to be very effective in the context of the exam paper. So I have seen some students who are very smart in the classroom setting. Whatever question I ask them, yeah, always have the correct answer. But sometimes in the paper, structured essay MCQ, they can make some mistakes. So that comes with practice. You have so much time left, and yeah, so we are going to keep doing this sort of exams. No need to stress out. If you are confused, you can ask me any question, right? And the only way is to go up, right? There's only progress from here, right? This is the first exam. Methane indala, you still have a lot of time, you still have a lot of potential, you still can practice yourself, right? You can still develop yourself, right? So as I always say, it's only upwards from here, right? දියුණුවෙන් විතරයි තියෙන්නේ. ඒක it will come to you, right? Only what you have to do is what? Hard work and work smart. Work smart and also put in the hard work, right? That's it. Okay. We can finish for today. Thank you for joining.